Are we, as this paper claims, one of the most important papers on the molecular mechanism of the detection of recombinant spike protein in the blood of individuals vaccinated against SARS-CoV-2. Here is the author in apparently Italy. So, of course, we go down to the methods section and ask, so how did he detect this recombinant spike protein in the blood of individuals vaccinated against SARS-CoV-2? And lo and behold, no surprise, probably, let's go down to the experimental procedures, informed consent. 20 human samples were collected from vaccinated subjects with informed consent. The geometric mean of their antibodies titer versus spike protein was such and such after 60 days. In addition, 20 human biological samples were collected from unvaccinated subjects and with informed consent. And so they were different. They were so-called, these ones who had not undergone COVID-19 and didn't have the vaccine and presumably had less tissue breakdown were negative for these antibodies, which according to them proves that the spike protein is created in the blood through vaccination and is part of the illness they're calling COVID-19. So again, the whole thing rests on the fact that the only thing that they measured here were antibodies. They were claiming that the antibodies were specific for the spike protein. Therefore, if they find the spike protein in the blood of vaccinated or people who allegedly had COVID, that means that they had spike protein disease, whereas the people who were healthy and then presumably not breaking down their tissues didn't have to make non-specific antibodies, so the antibody tests were negative. It has nothing to do with spike proteins or viruses at all. So again, it doesn't mean that I'm saying we're talking the mechanism here, not whether some people who allegedly had some non-specific illness called COVID-19 were sick. Maybe they were, and maybe they were breaking down. And I'm not exonerating these injections. For sure, if you inject somebody, as we'll see, with non-specific cell culture goop, you will make them sick, their tissues will break down, and they will have increased antibodies. The question we're dealing with here is not whether things can make people sick or injections of poisons can make people sick. It's whether the antibodies prove that this is a spike protein or a spike protein coming from a virus, and the spike protein is made by the alleged mRNA in the injection. So let me just go through, let me go through the next one here. So another big study that people sent me and wanted to know about, doesn't this study, circulating spike protein detected in post-COVID-19 mRNA myocarditis. You see, the tendency here, especially amongst the so-called freedom community, is they like to pick up on these studies to attempt to demonstrate or prove that these vaccines, so-called, are horrible and they're causing myocarditis, and they're doing so through the mechanism of this creation of this so-called spike protein. I am not arguing against the fact that the injections are horrible or that they give people myocarditis or otherwise heart problems. I'm talking about the mechanism because the mechanism is everything. It has to do with eventually how you think about this whole thing, what is actually happening, and even eventually how to treat it. Because I have no sympathy for the argument advanced by so many doctors. Tom, what difference does it make whether there's actually mRNA in the injections, or whether there's spike proteins, or whether there's a virus? It makes all the difference in the world, because if you can't understand what's happening, or at least disprove that this particular thing is happening, you will eventually be led astray. You will also eventually scare and frighten people more than you should. And there is no benefit from being ignorant about what happens and using anti-scientific thinking to make claims about what's happening that are easily disproven. So when you say, okay, well, how did this paper that's so crucial to our understanding that it's the spike protein that's causing myocarditis, how did they detect the spike protein? And no surprise there, if you go to the methods section, you see we performed extensive antibody profiling, and then there's a whole other bunch of immune profiles, antibodies against the human relevant virome. These are all downstream antibody testing, all of which are non-specific and can't possibly tell you that there was a spike protein. And here again, you see this immunophenotyping, and it's all about detecting antibodies against previous infection, SARS-CoV-2, spike protein specific T-cell responses, and other antibodies. They never actually assay for spike protein directly in the fluids. They sometimes look for pieces which they allege through other antibody testing previously done, that those come from the spike protein. It's all basically boils down to, are antibodies specific? And the answer, as I said, is clearly no. So this brings up another interesting question. So somebody could say, okay, Cowan, how can you actually go about proving whether these antibodies are specific or not? Like, what should we do? Just like we outlined with how they should go about proving there is a virus or not with our viral challenge, here I will outline how you would go about, if you wanted to do proper, reasonable, logical science, proving that antibodies are specific and not just non-specific reactions to tissue breakdown. So it would go something like this. You would give a substance, preferably a toxic substance or a substance that causes damage, like a vaccine, so-called, or an injection, or some sort of cell culture goop or nanoparticles. And then you would get breakdown of the tissue. If you don't give any toxic substance, you won't get any tissue breakdown, presumably, and then you won't get any antibodies produced, and then you don't have anything to study. So you give the substance, you get the tissue breakdown, and then you then you inject the antibody, or takes a sample and mix it with the antibody that you believe, this is what you're going to test, is specific for a certain protein. They say that if this antibody binds and therefore makes some sort of reaction, that's proof of specificity. But what they should do is give the same person or animal a different substance that couldn't possibly have a spike protein in it, but is also toxic to the tissues and causes a similar amount of tissue damage. Then, once you get the tissue damage, you take a sample or inject the antibodies or mix it with antibodies in the sample, or inject the antibody into the person and see if it binds the same antibody. If it binds, and obviously the insult, the toxin was different, that proves that the antibodies are not binding to a specific toxin. They're binding to non-specific toxins, and in particular, they're being produced in reaction to tissue damage. So that's the first of two controls that you would do. The second is you would give this toxic substance, let's say something you claim is a spike protein or an mRNA, you would see the tissue damage, and then you would inject it with the antibody that you claim is specific, see if it binds and see if it lights up and you can detect it. And if it does, you claim that binding proves that it's that protein specific. But then you give the same substance, your so-called spike protein, you get the tissue breakdown, but this time you inject or mix it with a different antibody, not the antibody that you say is specific to the spike protein, but a totally different antibody. That, of course, shouldn't bind, and if it does, it tells you that antibodies are binding non-specifically, and you cannot use it to prove the existence of that antigen or that protein in the first place. Every single paper that does that uses antibodies to make this claim, should obviously include both of those steps, and yet none of us can find a paper that ever includes both of those steps. Therefore, they are all anti-scientific. They are not using appropriate controls and not following the scientific method. And this is why one of the world's leading authorities on antibodies, and particularly monoclonal antibodies. Monoclonal means they're specific to one antigen, and that's Clifford Saper, Harvard Medical School professor. And this is a quote from one of his papers, quote, "No, there is no such thing as a monoclonal antibody that, because it is monoclonal, recognizes only one protein or only one virus. It will bind to any protein having the same or similar sequence. So there is no such thing as a monoclonal or antibody specificity." So all these papers alleging that they found the spike protein, that the spike protein is a mechanism of damage, need to be tossed out as uncontrolled anti-scientific garbage. If you want an analogy, I came up with one just before this that may help. So let's say you have a balloon and you cut the balloon with a knife or some round object, and then you put duct tape on it to fix the balloon. And then you claim that because you were able to fix the balloon with duct tape, this proves that the knife was the mechanism that cut the balloon. That's essentially what they're doing. They're saying essentially that the duct tape is somehow specific to the mechanism of injury, which is a knife. So the first control experiment you would need to do is take the balloon and cut it with a scissors, and then use your duct tape and see if you could fix the balloon. Because if you could, this would demonstrate that your conclusion originally was wrong, that it is not specific to a knife, because it works just as well with a balloon cut with a scissors. And then the next control experiment you would do is you would take the balloon and you would cut it with a knife, but this time you would try to fix the balloon with, say, elephant tape. I'm not sure what that is, but I've heard that that actually works sort of like duct tape. And if that works to fix the balloon, which it would, that would tell you that the type of tape, i.e. the antibody, is not specific to the mechanism of injury, that is to say a knife, that any similar tape would work. So again, similarly, many antibodies will bind to that protein or to that injured tissue because the antibodies are not specific to the tissue, to the protein, they're specific to the tissue injury. So many different mechanisms of injury and many different antibodies will work. And if you don't believe me, send me a paper where they did both of those controls, and I and others will admit we're wrong, except that won't happen because none of the so-called scientists will be able to do that, because as far as we can see, it doesn't exist. And so once again, we are putting out very specific guidelines to prove us wrong. And the people who are attempting to do that seemingly never are able to do that because those papers don't exist. And then finally, we get to the issue of Dr. Koresh's paper of, do these antibodies, is there actually mRNA in these injections? So here's the paper. You can see the reference here, and I don't know exactly how to find it, but I think if you put this in, somehow you'll be able to find it. And he talks about how they claim that there is mRNA in these injections. I mean, that's the whole point. You put the mRNA for the spike protein, then that goes to the imaginary ribosomes and makes the spike proteins, and the spike proteins make non-specific antibodies to a protein that couldn't possibly have been made, or at least has never been demonstrated to have been made. And pretty soon you realize you're in la-la land. So here he goes through the steps. And I think basically he talks about the fact that the mRNA, let's just read it and so we'll go from there. From a pharmaceutical perspective, one must obtain the active ingredient, in this case mRNA. Either have to make it yourself or get it from a third party. So he talks about there's the active ingredient, which is the mRNA, and then there's all the other stuff that goes into the formulation. So we're not interested in the other stuff. We're only interested in this so-called active ingredient, which is mRNA. So during the product development, the active ingredient is monitored, tested to see if it is in the body as expected and the expected amounts. The efficacy and toxicity relate to the active ingredient levels. Therefore, a vaccine developer would first need an appropriate mRNA or its source to purchase. Such an active ingredient should commonly be available from an independent third-party supplier with appropriate certification for identification and purity. However, the COVID-19 mRNA is proprietary. No information about its nature and purity is available in the public domain. So obviously that makes it difficult to know whether that's in there. Therefore, as he says appropriately, one must rely on general information regarding what is present in the vials and how they may have been synthesized, manufactured, and purified. So now we're getting to the crux of the matter. In this regard, a fermentation process using culturing microbes, such as bacteria, is claimed to produce mRNA, which is then extracted isolated. From the manufacturing perspective, the following diagram shows the steps. You can see that the steps, hard to see here. The culture is developed, some chemical reactions are performed. This stops the culturing fermentation, followed by purification. The last step is marked as formulation. This production process of mRNA is simple yet very confusing, which may be why people do not correctly understand the manufacturing of the vaccine and its adverse effect. As explained above, the active ingredient is mRNA, and this is the key of all this. But no step describes mRNA production. If you go through this in detail, there is no step proving that this bacteria in this fermentation mat are making a specific mRNA. The last step in the diagram is formulation or vaccine. Therefore, this is vaccine production, not mRNA per se. He says they use the words mRNA and vaccine interchangeably, which is incorrect. Calling the end stage as formulation indicates that the mRNA has never been produced, but is assumed to be there. So there is no step in here that proves or demonstrates the specific production of mRNA. It's only assumed to be there. The last step in the manufacturing should be a pure and isolated mRNA compound. However, it is an isolate, quote, culture or gunk, possibly selectively concentrated compared to the one in the productive chamber. In other words, all they have is the breakdown of the culture or gunk, not specifically isolated, purified mRNA, which then they could use as the active ingredient to put into the vials. And he says they don't appreciate the difference between culture isolate gunk and pure isolated component, which is a critical misunderstanding as the relevant science, the same as the virus issue. So mRNA has not been produced, but a culture isolate gunk is considered and sold as mRNA or vaccine. And this is another crucial point he makes. It may be argued that the manufacturing processes or steps shown in the figure above have multiple filtration, separation, or isolation steps, like gradient ultracentrifugation for virus isolation, ensuring the production of pure mRNA. And this is the part that I can't verify myself, but I know Saeed, and I think this is a worthy place to start. Considering my extensive expertise and experience, 40 plus years in separation science, including exhaustive training and experience in chromatography, I can confidently say that the steps described here would not be able to produce the claimed pure and isolated mRNA until shown otherwise. Another critical point is it impossible to monitor mRNA production because no test may be developed without the availability of the pure and isolated reference standard. Therefore, it is safe to conclude the mRNA production is based on assumption, not scientific or valid testing. In other words, if they can't come up with the pure isolated mRNA, there's no way to validate this procedure, and therefore there's no way to claim that these vials actually, or this procedure made the mRNA that they're saying is in there. Therefore, there's no way to even know that the mRNA is in there. So what's in there? He suspects that the presence of DNA contamination, which is becoming an issue now, they know that the DNA is contaminated, is simply because they're using cultured gunk or chip particles of bacteria, which obviously have their own contaminating DNA. And this contamination would explain the widespread adverse reactions after the injection of these vials. So we don't need to propose a mechanism of mRNA or spike protein simply injecting bacterial cultured junk with all the stuff that's in there that is not properly purified, and there's no way to assess the validity of the claim because they don't have a pure mRNA to begin with, makes the whole burden of proof on the manufacturers to prove that there is the mRNA that they say there is in there. And my guess is that's, again, a challenge that they will never undertake due to claims of proprietary or this or that, or we don't want to sell our secrets, or people would do nasty things with it as if they're not doing enough nasty things with what they're doing already. So again, there is no actual clear scientific evidence that this process would result in pure mRNA of a specific type that could be put into these vials, that could produce a spike protein, and that could be the saving grace of the pharmaceutical industry with further mRNA vaccines. It's simply the old culturing, non-specific stuff that they've been doing all along with viruses, and claiming they're actually doing something a lot more sophisticated than they actually know how to do.